Bismillahirrahmanirrahim, starting with the name of Allah, the most beneficent and the most merciful. Hello everyone, you are watching me Zahur Islam official. Today I will discuss you something special regarding the nanotechnology and biology and medicine. Now you can see here on my screen. So first of all, I would like to introduce about the nanotechnology. So as we know that in nanotechnology, the meter, a dimension unit closest to the everyday human experience, is often considered is the basic dimension of reference for human beings. Let us look up in the dimension scale of to the outer edge of our local universe, the Milky Way, a galaxy of 100 to 400 billion stars. This universe revealed to us has a dimension of 50,000 light years from the outer edges to its center. So, in the same case, a light year is the distance that a light travel in 1 year at the speed of approximately 300 millions miles per second, which correspond to approximately 10 raised to power 16 meter. So, therefore, the distance from the center to the outer edge of the Milky Way is 5 multiplied by 10 raised to power 20 meter or mile. So, let us know, look down in the other direction of the dimensional scale down to a nanometer, which is a billions times smaller than a meter. So, for example, 10 raised to power minus 9 meter is a nanometer or nanoscale. So, the word nano is derived from a Greek word meaning dwarf. So, in dimensional scaling, nano refer to 10 raised to power minus 9. For example, 1 billionth of a unit. A human hair has a diameter of approximately 10 micrometer, which is 10,000 nanometer. So, it means that the diameter of the atoms are in the order of the 10 raised to power minus 1 of nanometer. So, where is the diameter of the DNA strand is about a few nanometer. Thus, the nanotechnology is a general term that refers to the techniques and methods for studying, designing and fabricating the devices at the level of the atoms and molecule. So, the initial concept of investigating material and biological system at the nanoscale dates to more than 40 years ago, which records Feynman presented a lecture in 1959 at the annual meeting of the American Physical Society at the California Institute of Technology, and this lecture entitled that there is a plenty of room at the bottoms is generally considered to be the first look into the world of material, species and structures at the nanoscales level. Thinking small, however, it is not a new idea. Thousands of years ago, the Greek philosophers Leucippus and Democritus have suggested that all matter was made from tiny particles like atoms. So, only now the advent of nanotechnology will lead to the development of a new generation of instruments capable of revealing the structure of these tiny particles conceived since the Hellenic age. So, it is now generally accepted that nanotechnology involves research and development on nanomaterials and species at the length and scales from 1 to 100 nanometer. As we know that nanotechnology is very important to biology, since many biological species have molecular structure at the nanoscale level. So, these species comprise a wide variety of basic structures such as protein, polymers, carbohydrates, sugar and lipids, which have a great variety of chemical, physical and functional properties. As it is clear that individual molecules, when organized into the controlled and defined nanosystem, have new structure and exhibit new properties. So, this structural variety and the versatility of these biological nanomaterial and systems have important implication for design and development of new and artificial assemblies that are critical to biological and medical application. The development of a next generation nanotechnology tool kit is critical to understand the inner world of the complex biological nanosystems at the cellular level. Since the nanotechnology involves technology and the scale of molecules, it is the potential of developing devices smaller and more efficient than anything currently available. Traditionally defined disciplines such as chemistry, biology, material sciences also deals with atoms and molecules, which are the nanometer sizes. But nanotechnology differs from the traditional disciplines in a very fundamental aspect. For example, chemistry or biology and material science deal with the atoms and molecules at the bulk levels. So, we do not see the molecules in chemical solutions, whereas the nanotechnology seeks to actually manipulate or individually atoms and molecules in a very specific way, thus creating a new material having new properties and new function. So, it means that nanotechnology play a very important role, especially in nowadays and research methodologies, that how to manipulate the individual atoms and a molecule and a specific sequence, that creating a new material having new property, having new function. So, it is this bottom up capability that makes the nanotechnology a unique new field of research of undreamed possibilities and potential. Or mastering of nanotechnology could unleash breakthroughs in genetic engineering, genomics and proteomics and medicines in the coming decades. If we can assemble the biological system and devices at the atomic and molecular level, so we will achieve the versatility and design, a precision and construction and control and operation. And here, so therefore, it is hardly imagines. Now, come toward the cellular nanomechanics and the building blocks of life. As we know that nanotechnology is one of the great importance to molecular biology and medicine, because life processes are maintained by the action of series of biological molecular nanomedicines and the cell machinery. By evolutionary modification over trillions of generations, living organisms have perfected an armory of the molecular machine structure and properties and process. So, the living cells with its myriads of biological components may be considered the ultimate nano factory. So, you can see here in figure 1.1 show a schematics diagram of the cell. So, this is the structure of cell. You can see here, this is the schematic diagram of cell and its components, or these are the cell organelles. So, look at the vesicles, look at the actin and myosin filaments, look at the intermediate filaments, and then mitochondrions, and then cell membrane, rough endoplasmic reticulum, then ribosomes, cytosol, Golgi bodies, smooth endoplasmic reticulum, centriole microtubules, nuclear membranes, nucleus, nuclear pore, lysosome and peroxisome. So, what does it mean? So, I mean that in table 1.1, this is typical nano sizes of cellular species. These are different type of species, and you can see the different sizes and its molecular weight which are given in the table. This is the biological species. Biological species are small assemblies. Small assemblies are ribosomes. You know, ribosome is one of the essential important component of the cell organelle. So, it has very typical size 20 nanometer sphere, and its typical molecular weight is 10 to 5 and 10 to 7. Look at the large assemblies, viruses. So, viruses is typical size is 100 nanometer sphere. So, it cannot be seen with naked eye, can be only observed with the help of microscope. So, the typical molecular weight is 10 raised to power 7, 10 raised to power 12. The nucleic acid is a biological species, and its example is transfer RNA. Its size is 10 nanometer rod and its typical molecular weight is 10 raised to power 4 to 10 raised to power 5. The small proteins, these are called chymotrypsin. Chymotrypsins are small proteins which are biological species, and it is typical size 4 nanometer sphere. Its typical molecular weight is 10 raised to power 4, 10 raised to power 5. The large proteins which are also biological species, that is called aspartate trans carbamylase, so that its typical size is 7 nanometer sphere and its typical molecular weight is 10 raised to power 2, 10 raised to power 7. These are the various components which are also given in the schematic diagram as well as in the table. So, some typical sizes of nucleic acid, proteins and biological species are shown in the table 1.1. Nucleic acid and proteins are the important cellular components that play a very critical role in maintaining the operation of the cell. So, as we know that the nucleic acid and proteins, so they play a very extravagant role in the cellular components, so that play a very critical role in maintaining the operation of the cell. DNA is a polymeric chain. As we know, the deoxyribonucleic acid, which is a polymeric chain made up of subunits called nucleotides, which are the building blocks of DNA. The polymer is referred as to polynucleotide, and each nucleotide is made up of sugar, a phosphate and base. There are 4 different types of nucleotide which are found in deoxyribonucleic acid in the structure of DNA, differing only in their nitrogenous bases like adenine, guanine, thymine and cytosine. The basic structure of DNA molecule is helical, with the bases being stacked on the top of each other. So, DNA normally has a double stranded conformation with 2 polynucleotide chain held together by a weak thermodynamic forces. So, 2 DNA strand forms a helical spiral winding around a helical axis in a right-handed spiracles. The 2 polynucleotide chain run in the opposite direction, and the sugar phosphate backbone of the 2 DNA strand wind around the helical axis like the railing of the spiral staircase. So, the bases of the individual nucleotide are inside the helix, stacked on the top of each other like the steps of a spiral staircase. Genes and proteins are intimately connected because genes are lying on chromosomes. So, the genetic code which is encrypted in the DNA is decoded into the corresponding sequence of the ribonucleic acid, which is then read by the ribosomes to construct a sequence of the amino acid, which is the backbone of specific protein. The amino acid chain folds up into a 3-dimensional shape and becomes a specific protein which is designed to perform a particular function. Ribosomes are important molecular nanomachines that build up proteins essential to the functioning of the cell. Although the size of typical ribosome is only 8,000 cubic nanometers, nm cubic. So, this nanomachine is capable of manufacturing almost any protein by stringing together amino acid in a precise linear sequence following instruction from a messenger RNA copied from the host DNA. To perform its molecular manufacturing task, the ribosome takes hold of the specific transfer RNA, which is turned and chemically bonded by a specific enzyme to a specific amino acid. It is means to grasp the growing polypeptide and to cause the specific amino acid to react with and to be added at the end of. So, at the end of the polypeptide, in the other words, as we know that DNA can be considered to be a biological software of the cellular machinery, because DNA is a living material, whereas ribosome are large scale molecular constructor and enzymes are functional molecular size assemblers.